So, Piotr, here we are in your lab in the depths of the Natural History Museum. I mean, we're in a really privileged place to actually come in and see this, because not many people get to see the stuff that goes on literally behind the scenes at the museum. Just talk me through, first of all, where we're at, what space we're in here.
Well, it's good to have you here, and I'm happy to show you around the backstage that, as you said, not everyone has access to. We are now in the molecular labs of the Natural History Museum, where we are performing all the research focusing on DNA and genes and genomes, sequencing the genomes and genes of wide variety of organisms and samples.
And we're specifically having a bit of a conversation around the Blean project and the impact that the bison are starting to have on the soil there.
The bisons will, the presence of the bisons and the more so like the effects of their activities will affect the soil diversity in a way that we expect more species of bacteria will appear that will help, for example, to store the CO2 in the soil. They should affect the diversity of the plants. This is most important. Higher diversity of the plants helps to store the CO2, right? And since the CO2 is being absorbed
CO2 is a big
plants,
issue at the moment, isn't it?
Yeah, and it's causing the climate changes, and plants are absorbing it through the photosynthesis. So the more plants we have and more diversity of the plants we have, the more carbon dioxide is being removed from the atmosphere.
And is it too simplistic to say that the more biodiversity you've got in the soil, the healthier the soil is, and the more they can...
Of course, the more plants can thrive in that soil, and more of them can store carbon. Bacteria themselves, different species should encroach the soil, because as I mentioned, as the bisons mechanically also change the structure of the soil, they introduce more air inside the soil, which helps different species of microbes that require oxygen for their activity to encroach. And actually those bacteria that also are capable of performing photosynthesis, they require oxygen as well, the presence of oxygen. So they will support this process even more.
So physically, the way the bison knock down trees and they have their dust wallows, and the way their hooves dig into the ground, all of that is part of it.
Exactly, this is the part of it. Not only bisons themselves, regarding airing of the soil, contribute to this. Moles, by digging their burrows and holes and channels in the soil, they help with this. Earthworms as well. That's why they are super important.
Because you, as a molecular biologist, are actually literally digging around in the soil and see what's going on.
Exactly, this is how it looks like. And I wanted to say that I'm very happy we are part of this project. This is a very exciting opportunity to do a good science, and actually to check how our great engineers of nature, bisons, actually affect the soil here in the UK and in the Blean forest. So exactly, that's how it looks like. We have to go into the field, and with our collaborators from the Kent Wildlife, actually they collected the first batch of samples for us. But we are hoping to participate in further collections as well, to go into the field actually, because us as molecular biologists, we are usually locked in the labs, but sometimes we are released and we can go out and participate in other parts of the sampling process.
Now, some people might be confused about what has molecular biology got to do with wilding, rewilding, conservation. So what are you actually doing? What's the point of the sampling?
Yes. So after the samples are collected and sent to us, we are processing them in a way that we can extract the DNA from all the contents in the soil, because, as you know, the soil is the source of a huge variety of organisms and biodiversity. Literally 25% of all the organisms rely somehow on soil, and only 1% of microorganisms has been so far discovered and known to science, and are known to science. So we might all be stepping on a whole bunch of new species that we have no clue what they're doing and what is their role in the environment and ecosystems. So with the molecular biology, it is easier to study the diversity of those microbes, because with classical methods where you have to take the sample and try to grow colonies of different bacteria, not all of them can grow in laboratory condition, and it's literally impossible to grow them. So they skip the attention, they are being missed. And with our methods, we extract the DNA from all the content from the soil, so all the bacteria, all the fungi that were present in the soil, and also fragments of bigger organisms such as plants, parts of roots, right? And also cells from bison, for example, can be present in the soil, right? And even human DNA can be found. So with this extraction method, we extract the total DNA from the sample, and then using special machines, we sequence it so we read the message encrypted in the DNA.
Okay, and you before we've actually started recording this conversation, you were showing me some of the kit that you actually use to you, and it's like proper Star Trek kind of stuff, isn't it? That you've got a reader that you can put a drop of soil sample into, and then it literally works out every single different bit of DNA it can find in that sample.
It's not as easy as that. We don't put the soil directly on it. First, it's a quite longish process. It can take up to a few hours to extract the DNA. Then you have to purify the DNA because soil contains lots of unwanted material, right? That could block all this technology and all the reader's sensors, right? So we need to get rid of it, and that takes some time. After that, we can load the sample of purified, just pure DNA directly on the sensors, on the machine. And this reader is called sequencer, the machine that deciphers the information.
And so in one, because you literally put a drop of purified DNA sort of source material in from how many different species or strands of DNA do you expect to get from one sample?
From soil sample, we are expecting to detect hundreds of species of different fungi, different bacteria primarily. It might be possible to detect as well bigger organisms such as earthworms, for example, or as I mentioned, the remnants of plant material. But that is quite unpredictable, especially if you don't see with your own eyes the parts of roots, right, of different plants. Then you can predict sort of, okay, we will see this plant in these results, right?
So part of the project is to see how things change over time, the kind of impact that the bison are actually going to have on the soil. Is that the primary bit that you're looking for?
Exactly. I believe the whole design of this experiment of Wilder Blean rewildering is to see the total impact of the bisons on the changes in biodiversity, and our lab is focusing on this one aspect of soil biodiversity because we want to check if some discrete changes in the diversity are happening when the bisons are present, and we want to see how the bison affects also the soil diversity, which you cannot see with your own eyes. You need to use this piece of equipment.
And it's very early on in the project, isn't it? You've only had sort of one sample, so you can't sort of say whether they're making a difference yet. So how long before we start to know what kind of an impact they're actually having on the soil?
Yes. So the first batch of samples that we received are our baseline samples, which were collected before the time the bisons were released. These samples will tell us about the current condition of the soil diversity in the area where the bisons were being planned to release. And now our aim is to collect the samples every 2 years to see when the bisons are present, and we'll compare them from the exact same locations and at the same time of the year to exclude the variability of the seasons and maybe the variability of the soil itself. So we'll collect those subsequent samples from exactly the same spots.
And for you as a scientist, as a molecular biologist, is this an interesting project? Is it an exciting thing to be part of?
It is a super exciting project for me, not only because of its scientific content. I really like the design, how the whole project has been designed, because it helps our lab as well to do good science. Probably it was mentioned before that the forest has been separated into 3 different regions where there are control areas. So actually thanks to this we can deliver a really rich content of results and conclusions, right? Whether the bison has any effect on the biodiversity. Apart from that, it's very emotional for me as well, because bisons themselves are the symbol of how we can repair the damage that we are doing to the environment. And also, I'm Polish, and Poland was one of the countries that contributed the most to saving this species. So it's very, very emotional for me as well.
Just tell me a bit about yourself. How did you come to be working here at the Natural History Museum?
To be honest, secretly it has always been my dream to work here, because for the type of biologist that I'm a combination not only of a molecular biologist but also classical biologist. I'm fascinated by zoology, and especially in taxonomy, by diversity of the species. And I always sort of dreamt working here. I was volunteering here as well for many years with some of the departments, and eventually it happened, and I applied for a job, and I was successful, and I'm here 3 years now.
It's an amazing space, isn't it? Because I certainly didn't realise quite how big the Natural History Museum is. You know, I've been in, I've seen the dinosaur skeletons. I used to come here as a child. I brought my kids here. But there is an amazing amount of work that the Natural History Museum is doing in real science terms all the time.
Exactly. We have a wide variety of projects, not only our lab, but all the researchers within the museum, which is around 200 staff. And they spread from studying the taxonomy of particular group of animals or plants, to studying environmental samples such as soil samples, or samples from the deep sea, from the permafrost as well, from remote islands, unreachable for people, from the depths of the ocean. So that's enormous amount of wide variety of science, and not only within natural sciences. I'm a biologist, so I can talk about the biological part of the research that the museum does. But we have enormous geological department, and also we are dealing with space as well, right? Some meteorites are being studied here, and soil samples from Mars, for example. That's what I know. And also we have a conservation team that deals with some science regarding conservation of our collection, right? This is also a science, how to preserve specimens, to prevent them, like taxidermy, from being attacked by fungi and being destroyed.
And presumably, if you're doing a particular bit of research on whatever it happens to be, and you need to talk to another expert, you can literally walk around the corner and find them here in the building.
Exactly. That's what we do. We communicate with each other, and now the departments of life sciences and earth sciences have been merged into one science department to enable this communication and make it even easier for us to collaborate with each other, because nowadays we cannot be experts on everything. It's literally impossible. The sciences are too big. We need to collaborate with each other, because like bricks in the wall or pieces of the puzzle, we complement each other, and then we can do even more science.
Wow, it's a fascinating project, and I'm really looking forward to seeing some of the results of it. So we'll come back in a couple of years, have a chat again, see what surprises have cropped up.
Exactly, and definitely we will show you some changes, because that's the nature of nature, which never stands still and keeps changing. The question is how the changes will occur and in what direction they will be going. After 2 years already, we would be able to say something, but the idea of this project is not only to last for those 2 years, so hopefully it will continue for many years, as long as the bisons will be there.
Well, that was an absolutely fascinating conversation with Piotr. I'm letting him get on with doing some important work. There's more than 200 scientists actually work here at the Natural History Museum, getting on with all sorts of fascinating stuff. I'm really looking forward to coming back here in a couple of years' time to have a chat with Piotr again, to see exactly what is going on with the soil at the Blean, to see how it has changed over time, what it is the bison are actually doing. Don't forget, you can keep up to date with all the stuff that's going on in Kent Wildlife Trust through the podcast. Just search for Kent Wildlife Trust, A Talk on the Wild Side, wherever you get your podcasts, and you can hear me chatting with all sorts of fascinating and interesting things. See you soon.